For 50 years. I mean, the smartest scientists on Earth thought they were fighting a virus.
Oh, completely. They were totally convinced.
Right. They were throwing everything they had at it, operating under this complete misconception, and honestly, just losing.
Yeah, they were looking in the totally wrong direction.
So today, we are looking at a microscopic home invader that, well, literally wires itself into your cell's power grid. It builds an invisibility cloak and somehow manages to remain entirely undetected.
It's terrifyingly good at what it does.
It really is. We are talking about a word you have definitely heard, but almost certainly do not understand, and that is chlamydia.
Right, which is a term burdened with just so much cultural baggage.
Oh, absolutely.
You know, it's usually related to awkward conversations at a clinic. Because of that, the sheer biological genius of this organism just gets completely ignored.
Which is a huge shame. And honestly, that is the mission for this deep dive today. We are peeling back all that stigma to look at an evolutionary masterpiece.
Yeah, it really is a masterpiece.
Because using a stack of microbiology research and medical history notes, we aren't just looking at this as a common STI. We are exploring a bizarre, almost alien survivalist, an obligate intracellular bacterium.
And that phrase right there, that is the key to everything it does.
Obligate intracellular.
Exactly. Obligate intracellular means this organism has backed itself into an evolutionary corner, right? It absolutely cannot replicate outside of a living host cell.
Wow. Okay.
Like you could drop it into a petri dish filled with the richest, most perfect bacterial nutrients imaginable, and it would
Just sit there.
It would just sit there. It requires a living host. It needs you.
Okay, let's unpack this. Because to fully appreciate the mechanics of how this thing breaks into our cells, we first have to appreciate how thoroughly it broke the rules of science.
It broke all the rules.
I mentioned earlier that it fooled the world for half a century. Let's start there. How does an entire global scientific community misidentify a bacterium as a virus for 50 years?
Well, we were essentially victims of our own technology, you know. In the early 20th century, microbiology was largely defined by a few blunt instruments.
Right, they didn't have the tools we have now.
Exactly. And one of the main tools was the Chamberlain filter. It's this unglazed porcelain filter with pores so incredibly microscopic that ordinary bacteria would just get stuck in it.
Like a coffee filter for germs.
Pretty much.
Yeah.
So if a pathogen was small enough to wash right through that solid porcelain, and if it completely refused to grow on those standard nutrient agar plates we just mentioned.
Scientists assumed it had to be a virus.
Right. And chlamydia just waltzed right through those
Like
filters.
it squeezed right through.
Yeah, it was incredibly tiny. And because it only replicated inside cells, the agar plates always came up empty. It ticked literally every single box for a virus of that era.
But even before they were calling it a virus, the visual evidence they had was just fascinating. I was reading about this going back to 1907. There was this massive public health emergency in Java.
Ah, yes. The trachoma outbreak.
Right, involving this blinding eye disease called trachoma. So you have a German physician, Ludwig Halberstatter, and a Czech zoologist, Stanislaus von Prawacek. They're taking scrapings from patients' eyes and putting them under a microscope.
And they were looking for the causative agent of the blindness. What they found inside the eye cells were these strange, really dense clusters.
Yet not just floating around, right?
Exactly. The clusters weren't just floating freely in the cellular fluid. They appeared to be wrapped up in a distinct protective membrane, like a mantle.
So they named it chlamydozoa, which the ancient Greek word chlemis refers to a short mantle or a cloak, specifically the kind worn by ancient Greek soldiers.
It's such a great visual.
It really is. The entire genus is literally named after this visual impression of a microbe throwing a cloak over itself to hide.
Which is remarkably poetic, you know, considering they were essentially looking at shadows on a wall. They just didn't have the molecular tools to see what the cloak actually was.
Yeah, they were guessing.
Right. And for decades, researchers kept hitting a brick wall trying to grow it, which of course only entrenched the mistaken virus theory.
Which brings us to 1957. And this is where the history gets a little, honestly, unhinged.
Oh, this story is wild.
So a Chinese microbiologist named Tang Feifan decides he is going to be the one to finally culture this thing. And he figures out that if it needs living tissue, he obviously can't use agar. He uses the yolk sacs of embryonated hen's eggs.
Which was a brilliant leap, actually. Embryonated eggs were primarily being used to cultivate known viruses like influenza.
So it was already an established virus tool.
Yeah, the yolk sac is a living, incredibly nutrient-rich, totally enclosed environment. Kang realized that to grow an intracellular parasite, you had to provide it with living cells to invade.
So he successfully isolates the pathogen from the yolk sacs, but then he encounters a problem. The scientific community is skeptical.
As they always are.
Right. They want proof that this specific egg-grown isolate is the actual cause of the trachoma eye disease and not just some random contaminant from his lab.
And to prove causation.
Wait, I just have to stop here. To prove causation, he literally inoculated his own eye with the isolate. Like, he actually did that.
He absolutely did. He took the bacterial culture he grew in the lab and intentionally infected his own eye.
I cannot fathom that level of dedication or madness. What happens to him?
Well, he developed a severe case of keratoconjunctivitis.
Which is bad, right?
Very bad. That is an intense, incredibly painful inflammation of both the cornea, the clear front surface of the eye, and the conjunctiva, the membrane covering the white part of the eye.
Oh, wow.
It's incredibly dangerous. He risked permanent scarring or even complete blindness. But by suffering through it, he provided absolute, undeniable proof of causation. His isolate was the culprit.
It's terrifying, but, I mean, Tang's sacrifice blew the doors off this mystery because once we could reliably grow it in eggs, we could finally study it.
Exactly, and in the 1960s, technology advanced, and scientists peered under that Greek cloak.
And what they found changed everything. They found both DNA and RNA, and viruses generally only carry one or the other, right?
Right, almost never both.
They also found bacterial ribosomes, and they realized it was sensitive to antibiotics, not antivirals. It was a bacterium all along, just living this bizarre dual life.
And that dual life is what we call a biphasic developmental cycle. It is the biological engine driving everything chlamydia does.
Biphasic meaning two phases.
Exactly. It alternates between two totally different physical forms to survive the two very different environments it encounters. You got the harsh world outside the host, and then the interior of a human cell.
And the two forms are the elementary body, the ED, and the reticulate body, the RB.
Right.
The best way to visualize this cycle, and I love this analogy, is to think of the elementary body as an astronaut in a heavily armored, rigid spacesuit.
That's a perfect way to look at it.
The EB is tiny, dense, and practically dormant. It has no functioning metabolism. It is built entirely to survive the hostile vacuum of space, or, well, in this case, the environment of a human throat, an eye, or a reproductive tract.
Yes, but once this armored astronaut lands on a susceptible human cell, it breaches the hull.
And once inside, it takes off the spacesuit, unpacking into the reticulate body.
Yeah, that RB is the fragile worker form that actually builds the factory and replicates.
It's just wild.
The spacesuit analogy is perfectly apt, especially because that suit is completely inert. The EB doesn't swim around looking for a target. It just passively waits until it physically bumps into an epithelial cell.
It's just drifting.
Right. But the moment contact is made, the inert astronaut wakes up and actively tricks the human cell. It reaches into the host and hijacks the cell's actin cytoskeleton.
And the actin cytoskeleton is basically the cell's internal structural scaffolding, right? Like its muscles and bones.
Exactly that. The bacterium manipulates those microscopic muscles, forcing the human cell's membrane to ripple, reach up, and pull the bacterium inside.
It tricks the cell into eating it.
Yeah, the process is called endocytosis. The host cell is essentially tricked into swallowing the burglar.
But normally when a cell swallows a foreign object, it dumps it into a lysosome, which is like the cellular garbage disposal filled with acid to destroy it.
And this is where the cloak comes in. As the bacterium is swallowed, it wraps itself in a piece of the host cell's own membrane, creating a custom-built vacuole. This is what we call the inclusion.
So it brings its own tent.
Pretty much. The bacterium immediately starts modifying this vacuole so it avoids the lysosomes entirely. It diverts itself off the cellular highway and sets up a safe room.
And inside that safe room, the spacesuit comes off.
Right. Within about 6 to 8 hours of entering the cell, the EB expands, loosens its rigid structure, and becomes the reticulate body.
The worker phase.
Yes. Now, if you pop the human cell at this exact moment, the RB would instantly die. It has no armor. But inside the cloak, it is completely protected.
So what does it do in there?
It begins dividing rapidly by binary fission. 1 becomes 2, 2 become 4. It divides relentlessly. And that inclusion vacuole swells up like a balloon, sometimes growing so massive it physically pushes the host cell's own nucleus out of the way.
It literally takes over the entire internal volume of the cell.
It really does. Then, once the factory has maximized its output, those worker RBs start packing up. They asynchronously put their spacesuits back on, condensing back into those heavily armored elementary bodies.
Getting ready to go back out into space.
Exactly. By 48 to 72 hours, the host cell is entirely compromised.
Wow.
The exit strategy is just as calculated. The bacterium basically has 2 options. It can simply cause the host cell to burst open, what we call lysing, releasing all the new EBs like a bomb.
Just exploding the cell.
Yeah. Or it can use a gentler, less destructive method called extrusion. The cell physically pinches off the entire inclusion vacuole, pushing it out in one big membrane-bound package.
Which is super sneaky.
Very. That package can then burst later, which delays the host's immune system from sounding the alarm immediately.
The biological efficiency is staggering. From one dormant little astronaut, a single infected cell can pump out over 1,000 new infectious EBs in a matter of days.
It's an incredible multiplier.
But doing all that heavy building, replicating, dividing 1,000 times requires massive amounts of energy. Where is it getting the power to run this factory without the host cell immediately dying of exhaustion?
Well, that brings us to the second pillar of its survival. Chlamydia is fundamentally an energy parasite.
An energy parasite.
Yeah. Over millions of years of evolution living exclusively inside other cells, it experienced extreme genome reduction. Its genome shrank down to roughly 1 megabase, making it one of the smallest of any freestanding bacterium.
It basically Marie Kondoed its own DNA. It threw out anything that didn't spark joy or wasn't absolutely necessary.
That's a hilarious way to put it, but it's true. It threw out the blueprints for its own power plant. Generating ATP, the universal energy currency of all living things, is a complex, resource-heavy process.
Right. It takes a lot of effort to make.
So why maintain the machinery to build your own ATP when you're swimming in a cytoplasm full of it?
Oh, man. So it literally steals the cell's electricity.
It does. It manufactures a highly specialized protein called an AB-pan-EP translocase. The bacterium embeds this protein into the membrane of its cloak, plugging directly into the host cell's internal fluid.
Like plugging an extension cord into someone else's wall?
Exactly. When the host cell generates fresh, energy-rich ATP, the translocase snatches it, pulling it inside the cloak, and spits out a depleted, spent ADP molecule in return. It wires itself directly into the host's power grid.
Okay, here's where it gets really interesting. Because if you set up an illegal factory in someone's basement and start draining their electricity to run your machines, eventually the homeowner's going to notice the massive power drain.
They absolutely will.
So to survive, you have to actively defend the factory from the homeowner. You can't just hide.
And that's exactly what it does. The cloak is far more than a physical barrier. It is an actively defended fortress, utilizing a mechanism called a type 3 secretion system, or T3SS.
I saw this in the notes. The literature frequently describes this as a molecular syringe.
That is the most accurate visual. It is a literal microscopic needle complex protruding from the bacterium. We see similar structures in other pathogens like Salmonella.
So it's armed.
Heavily armed. Chlamydia uses this syringe to physically punch through the wall of its cloak and inject bacterial effector proteins directly into the host cell's cytoplasm.
So it's sitting inside its bubble, stabbing needles out into the cell and pumping proteins into the surrounding fluid. What are those proteins doing?
They're executing a highly coordinated sabotage operation with 3 main objectives. First, chlamydia cannot manufacture all the lipids and cholesterol it needs to keep expanding that massive cloak.
It has to steal those, too.
Yep. So the injected proteins intercept host lipids that are naturally passing by from the cell's manufacturing center, the Golgi apparatus. It hijacks the host's supply chain, rerouting building materials to its own walls.
Supply chain theft. Incredible.
Second, and arguably most important, these proteins block apoptosis.
Which is the cell suicide, right?
Exactly. Apoptosis is the biological self-destruct sequence built into our cells. If a human cell senses it is deeply infected or irreparably damaged, it is programmed to fall on its sword. It destroys itself to prevent the infection from spreading.
The ultimate sacrifice for the good of the body.
Right, but chlamydia cannot allow that. If the host cell dies prematurely, the fragile RBs inside will die with it. So the injected effector proteins essentially cut the wires on the cell's alarm system, suppressing apoptosis.
It forces the human cell to stay alive.
Yes, it forces it to stay alive and functioning while being drained of its energy. And third, these proteins actively suppress the fusion machinery that would normally send those destructive lysosomes to dissolve the inclusion. The factory is defended on all fronts.
This level of manipulation is stunning, but it's also the reason chlamydia created such massive blind spots for scientists, isn't it? Its integration into the host cell is so complete, it led to decades of scientific paradoxes.
Decades of confusion, yeah.
The most famous being the chlamydial peptidoglycan paradox. Wait, let me get this straight. How are we prescribing penicillin for decades if we couldn't even prove the target of the drug existed?
It was an embarrassing gap in our knowledge, honestly. Penicillin and related antibiotics work by destroying peptidoglycan, which is a rigid polymer mesh that forms the cell wall of most bacteria. It's their armor.
Right.
Doctors knew clinically that if you gave a patient penicillin, the chlamydia infection would halt. The drug worked.
But when they looked for the armor using every standard laboratory stain and extraction method,
They found absolutely nothing. Zero peptidoglycan.
that's crazy.
It ignited fierce debates. Some microbiologists argued the bacteria must have a totally different, undiscovered cell wall structure, and the penicillin was somehow accidentally hitting a secondary target we just didn't understand.
And we didn't solve this until 2013. I mean, in the timeline of modern medicine, that is essentially yesterday.
It really is. The mystery was finally cracked using a revolutionary technique called click chemistry.
How does that work?
Well, researchers designed modified fluorescent sugar molecules. These sugars acted like specialized Lego bricks that could only be picked up and utilized by the specific enzymes that build peptidoglycan. They fed these glowing bricks to the infected cells.
And because of the click chemistry, if the bacteria tried to build the armor, those specific bricks would click into place and literally light up under a microscope.
Precisely. And when they watched the cycle, they finally saw it. A tiny, incredibly thin, transient ring of glowing peptidoglycan.
Just a ring.
Yeah, it didn't cover the whole bacterium, and it didn't last. It only appeared for a split second, forming a ring exactly where the reticular body was actively dividing in half, and then it vanished.
It only builds the wall for the exact microsecond it needs structural support to divide, and then dismantles it so there's nothing for our immune system or standard lab tests to detect.
It's brilliant. An amount of armor so minimal and timing so brief, it evaded every classical method of detection for over 50 years.
That is mind-blowing. But the scientific community's blind spots didn't end there, did they? We also have to talk about the 1990s heart disease hypothesis, because this was a moment where the entire medical world thought they had just found the Holy Grail.
Oh, this is a classic cautionary tale.
Yeah.
It's taught in epidemiology courses today. The medical community was desperately looking for new ways to combat coronary artery disease, heart attacks.
The leading killer.
Right. And researchers noticed a striking pattern in serological studies. Patients who suffered from severe heart disease were significantly more likely to have circulating antibodies against chlamydia pneumonia, which is a respiratory cousin of the sexually transmitted strain.
They didn't just find antibodies, though, right? They found the actual bacterial DNA physically lodged inside the atherosclerotic plaques clogging people's arteries.
They did,
Yeah.
which created a perfectly logical mainstream hypothesis. Heart attacks involve immense inflammation in the blood vessels, and chlamydia is an expert at causing chronic low-grade inflammation.
So they put two and two together.
Exactly. The theory was, maybe heart disease isn't just about bad cholesterol and diet. Maybe this hidden intracellular bacterial infection is the match starting the fire in our arteries.
The implications of that are staggering. It meant we could theoretically prevent the leading cause of death in the world simply by treating high-risk patients with a cheap round of antibiotics.
The excitement was palpable. The medical establishment launched massive, multi-center, randomized clinical trials, most notably the WIZARD and ACES trials. They enrolled thousands of heart disease patients and gave them long courses of azithromycin.
Hoping to cure heart disease.
They fully expected to see a dramatic drop in secondary heart attacks and cardiovascular events.
And the result was, what, absolutely nothing?
Zero clinical benefit. The trials were a colossal disappointment.
Wow.
The patients taking antibiotics had the exact same rate of heart attacks as the placebo group. It proved that the bacteria in the plaques were likely just innocent bystanders, perhaps carried there by immune cells scavenging the blood.
So it was just a coincidence.
Yes, it cemented the golden rule of science. A strong, reproducible statistical correlation does not equal causation.
Okay, so it didn't cause heart attacks, but we cannot underestimate the damage it actually does cause, primarily because of how silent it is. You might think, if a bacterium has broken into my cells, set up a factory, and is actively draining my power grid, I am going to feel sick.
You would think so.
But that is the terrifying genius of this microbe.
We really need to clear up some dangerous misconceptions here. People frequently group chlamydia and gonorrhea together.
Right, because they're screened together.
Exactly. They are both common STIs, they are screened for at the same time, and they can produce similar symptoms in extreme cases. But biologically, they aren't even playing the same sport.
Gonorrhea is caused by a diplococcus. It looks like a pair of microscopic coffee beans, right?
Yeah.
It is free living. It doesn't need to hide inside your cells to survive. It thrives in the extracellular spaces. Because of that structural difference, they require totally different classes of antibiotics.
And more importantly, the way they cause disease is fundamentally different. Which brings us to the most critical point. A huge percentage of genital Chlamydia trachomatis infections, especially in women, are completely asymptomatic.
The patient feels absolutely fine.
Yes, but the lack of symptoms does not mean there is a lack of damage. And the irony here is tragic. We normally assume the bacteria itself is releasing toxins or eating tissue. But in this case, the bacterium just wants to quietly steal ATP and stay hidden in its cloak.
So our own body is the wrecking ball.
Precisely. This is known as the immunopathology model. When the bacteria is stressed, perhaps by a mild immune response or a lack of nutrients, it stops dividing. It enters a persistent dormant state inside the cell. It just lies low.
Hiding out.
Right. But your immune system is smart. It senses that something in the tissue is fundamentally wrong.
So it calls in the cavalry. It initiates a Th1-driven immune response. The body sends specialized white blood cells to the area, dumping inflammatory chemicals to destroy the perceived threat.
Yes, and I like to think of it like a military shelling its own city. To take out a sniper they know is hiding in one of the buildings, but they don't know which one.
Wow, that's a brutal image. The sniper isn't blowing up the buildings. The collateral damage from the constant shelling is.
Exactly. Over months or years, this repeated chronic inflammation destroys the delicate architecture of the surrounding tissue. In the reproductive tract, this causes severe scarring. This is the mechanism behind pelvic inflammatory disease, or PID.
And the tissue of the fallopian tubes gets so scarred and rigid that an egg can't pass through. It leads directly to ectopic pregnancies, which can be fatal, and preventable infertility, all happening entirely under the radar.
It's devastating, and the global impact is staggering. Beyond the millions of genital infections, different strains cause different devastating issues. Trachoma, which we mentioned earlier, is still the leading infectious cause of blindness in the world.
Still today. The repeated Th1 inflammatory response in the eyelid causes the tissue to scar and contract, physically turning the eyelashes inward so they scrape against the cornea until the patient goes blind.
That sounds horrific.
It is. The World Health Organization fights this with the SAFE strategy: surgery to fix the eyelids, antibiotics, facial cleanliness, and environmental improvement.
You even have Chlamydia psittaci, a respiratory strain carried by birds, which poses a serious hazard for poultry workers and pet store employees. With all this global damage, the obvious question is, why don't we have a vaccine? We can vaccinate against incredibly complex viruses. Why not this?
Because of that exact Th1 inflammatory response we just discussed, getting infected naturally doesn't give you lasting, sterilizing immunity. People catch it over and over again. Designing a vaccine like the CTH522 candidate that recently completed phase 1 trials requires navigating a brutal immunological tightrope.
Meaning you have to train the immune system to recognize and kill the bug without triggering the massive inflammatory cascade that causes the scarring in the first place. Right. If the vaccine inadvertently triggers a strong Th1 response, the vaccine itself could theoretically induce the very tissue damage and PID it's trying to prevent. You have to eliminate the sniper without shelling the city. We are still struggling to find the precise molecular signals to achieve that.
So what does this all mean? We started by talking about a routine clinic diagnosis, but what we actually found was an evolutionary titan. Chlamydia is an energy-stealing, cell-hijacking parasite that builds an active fortress. It acts so much like a virus it fooled generations of scientists, and it synthesizes its armor for a split second just to remain invisible.
It is the ultimate ghost in the cellular machine.
And looking at how perfectly it operates leaves a really lingering question. Given how flawlessly chlamydia has evolved to evade detection, manipulate our cellular machinery, and cause silent long-term inflammation, it makes you wonder, how many other mysterious, idiopathic, chronic inflammatory diseases out there, the conditions doctors simply cannot explain, are actually just the fingerprints of other obligate intracellular microbes we simply haven't learned how to see yet.
Our understanding of disease is only as good as the tools we use to look for it. The dark matter of microbiology is vast.
Something to mull over next time you wonder what might be hiding in the dark. Thank you so much for joining us on this deep dive. We'll see you next time.